Everybody ready? Good evening, everyone, and welcome. My name is Kevin Pitts. I'm the Dean of the College of Science, Virginia Tech, and I'm delighted to welcome you to the J. Mark Sowers Distinguished Lecture Series in the College of Science. We are very pleased to have you join us today for this special talk by Dr. Ronald Vale. The J. Mark Sowers Distinguished Lecture Series was initiated in February of 2017 with the goals of exchanging new and innovative ideas in science and providing opportunities for the university community and the general public to interact with eminent scholars. Thanks to the generosity of Mark and Debbie Sowers, and Mark is here with us today. We certainly appreciate him joining us. The sponsors of today's event and this lecture series, we're able to do this not only within the college, but also to offer these ideals and inspirations to the larger community. This event's special, and it's the first live event that we've had in 3 years. The last live event was the fall of 2019, and I think you all know why, but we are absolutely thrilled to be back in person, but also having a number of folks join us virtually through Zoom, both in person and a webinar format, so we can expand our audience to alumni and friends across the Commonwealth and around the globe. So on behalf of the university, I would like to thank Mark and Debbie Sowers very much for their tremendous support in creating this lecture series. It's absolutely fantastic. Their passion for science and discovery has made it possible for us to bring 15 distinguished scholars now and especially exceptional speakers to the Virginia Tech in recent years. So now I will invite Dr. Janelia Cimini, Professor of Biological Sciences, to come forward and introduce our speaker, Dr. Ronald Vale. Janelia.
Okay, I'm really honored to introduce Ron tonight. I ran into Ron at MBL in Woods Hole, where he spent many summers, and I'll tell you in a minute more about that. So Ron, as you all know, he's the Vice President of the Howard Hughes Medical Institute and is the Executive Director of the Janelia Research Campus. Now, but he spent many years at UCSF, where he's now an emeritus professor. He started at UCSF in 1987 after getting his PhD in neurosciences at Stanford University. We were just talking about that at dinner. And then he spent a couple of years at the MBL. So Ron has very long-term bonds to that place, and that's where he actually made like a lifetime discovery of the Kinesin motor that I think he's going to tell you about today. I am not going to tell you about all the big findings, research finding that he's made, because that would take too long. He's contributed to the field of cell biology tremendously. And a lot of what we know about molecular motors in cells and what they do and how they function is really owed to his discoveries. And like I said, he started in neurosciences, but he became a cell biologist, and one that we all look up to. So Ron is also a member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Science, the European Molecular Biology Organization, and the Indian National Science Academy. And he's received many prizes and awards, and I'm not going to read the list. I actually have the list, but I decided I'm not going to read the list because I would like to spend a minute or so to say something more personal. But the one award that I would mention is the Lasker Award in Basic Medical Research, which I compare to a Nobel Prize pretty much. It's one of the highest honors that you can earn as a scientist. But I think that what inspired me to nominate, so Silke Hoff, who's in the audience, and myself nominated Ron for this lecture series. And one thing that really inspired us is that Ron has done a lot for the scientific community. It's not just his science and his papers that have really impacted us, but it's really what he's done in terms of outreach and initiatives that they started and really moved forward. And one that we discussed at dinner also is the iBiology, which is a nonprofit organization that produces videos of scientific talks presented by leading scientists. And actually, I want to welcome Karen Dell, who's Ron's wife, and is also come here to visit with us. And I learned that she was actually instrumental in putting all those videos together. So good, really good partnership. Ron, you found a good partner there with this. And Ron did many other things. He founded XBiome, which is a kind of a learning resources for undergraduate biology. And he did a lot. So I kind of stress this Indian National Science Academy because Ron actually has strong connections to India, where he founded this India Bioscience, which is a networking organization for the life sciences in India. So he started this annual young investigator meeting for young Indian scientists, and also founded and directed a microscopy course in Bangalore, India. So he's really gone off and done amazing things for the community, but not just the community locally, like in the US, but also abroad. The other thing that I would like to mention is that his lab developed this free open source software for like microscopy, Micromanager, which is another thing that, like another example of how he's really impacted the community. And the last thing I want to say, so I mentioned that Ron spent some time when he was a young scientist in Woods Hole, and I know that that's a place, that's actually where I met Ron initially, because my postdoctoral advisor was going to Woods Hole every summer with part of his group, and they would all, you know, talk and run into each other downtown. And for me, I didn't necessarily interact with Ron much at that time, but of course there were people like him, and I'm glad to see so many students here, because there were people like Ron and others in Woods Hole that really inspired me to do this, you know, to keep doing this for the rest of my life. And then the last thing I want to say is that Ron actually in Woods Hole, and you know, in terms of his connection to the place, he also co-directed the physiology course, which is kind of a historical thing that has been going on for many years. So with Tim Mitchison for 5 years. And so the directors turn over every 4 or 5 years. And I think that what they did in terms of the physiology course was really to transform it much more into an interdisciplinary kind of course, where they brought in not just the traditional, it was microscopy initially, but they brought in mathematical modelers, which I think here at Virginia Tech we really like. we have a strength in that area, and biophysicists. And I think that's one of Ron's strengths, that is really always interacted with so many people in other disciplines. And it's a really good example on how you can answer questions if you look at that question from different angles. And I will stop there because I could go on and on, but I want to give him time for his talk. Thank you everybody for being here.
Okay, good. Daniela, thank you so much for that very warm and generous introduction. It was very kind of you, and Mark and Debbie Sowers for your inspiration of creating this series. And it's wonderful to be here. As you heard, my wife, Karen Dell, and I just moved to Virginia relatively recently in 2020. And when we came here, we were very eager to meet our neighbors, like Virginia Tech. And we've had the pandemic in between, but I really appreciate this invitation. And I hope this is at the beginning of a connection with Virginia Tech. So it's a great pleasure for me to come here today. This is where I work now, Janelia Research Campus. It's the research campus of the Howard Hughes Medical Institute in Ashburn, Virginia. And I'll tell you a little bit I'm the executive director of this campus, and I'll tell you a little bit more about Janelia at the very end of my talk. But the main subject of my talk today is about biological movement. Now, this is an attribute of life that everyone can appreciate. We are all familiar with motion in the natural world, like this beautiful cheetah that's moving. And this is examples of macroscopic motion. But when you look under a microscope, you also can see this whole other world of motion, cells moving around in their environment. And if we look even further, not just at the cell, but at the interior of the cell, as I'll tell you about later, we can also see, now we're looking inside of a cell, all kinds of objects moving back and forth, traveling within the cell. And this will be a main subject of my talk today. And in addition, if you watch a cell divide, and what you're looking at here in green is the DNA. This is a fly embryo, and this is the genetic material. This is the DNA. And you can see this DNA physically separate from one another as cells divide. So that, again, is an example of cell motion. Basically, wherever you look in biology, you find that movement and motility is fundamental to life. And this is the main subject that I'd like to talk to you about today. So let's start at the origin of some of the early discoveries of cellular motility. So this is a gentleman that many of you probably have heard of, a Dutch inventor and scientist, Van Leeuwenhoek. And he developed a new microscope, which is shown over here. It's actually very small. It consists of one lens. It's probably about this big in size. And he put it up to his eye. And with this very special lens, he looked at various things. He looked at everything, basically, including pond water. And what he saw, imagine this. Imagine for the first time seeing this invisible world and seeing this amazing creatures that no human being has ever seen before, also engaged in movement. And in any drop of water in pond water, you would find it teeming with life and moving life. And what he wrote is the motion of most of them in the water was so swift and so various, upwards and downwards and roundabout, that I admit I could not but wonder at it. Too bad we don't write scientific papers like that anymore. And here's a later example of another individual, Bonaventura Corti, who took a microscope and now peered not just at the cell, but at the interior of cells. And he saw something incredible. This is not his movie, but it is an example of what he saw, which is motion of chloroplasts inside of plant cells. And again, look at what he wrote. This is the first time anyone had ever seen this. I know that the phenomena that I announce is too strange to be believed at first. I saw two torrents inside each section. One of the torrents rose on one side and descended on the other constantly. And this not once, but thousands of times, and for days and for entire weeks. So you can again imagine, you can kind of feel this, the excitement of discovery. And that is partly what I want to share with you today, is what it means to discover something and how discoveries come about. And there is something truly amazing and remarkable, and as a scientist, I would say also very contagious and infectious, that seeing something or understanding something new about the natural world, something new that you've found for the first time that no other human being has ever derived insight into before. And, you know, I think this is what drives scientists and makes this profession so remarkable. Now, I also want to contrast this a little bit between, a little bit what we teach students, what the scientific process is, and a little bit what the ground truth reality is. So, this is something, a page that I just took from a textbook. You don't have to go through all of the details here, but it seems like this very stepwise process and, equation to what scientists do and how discoveries are made. And, to simplify it a little bit, it kind of comes across, I think, often to students that the way the scientific process works is that, first of all, you just sit around, you think of something really brilliant, and then you design a really well-designed experiment to prove that thought. And then you celebrate how smart you were, that the experiment worked. And I'm going to give you a different view of this idea of what science is and how it works. Of course, this view here, you think of, brilliance, and you think of someone like Einstein, and of course, Einstein was, really brilliant, but I think there's more room for other ways of thinking about science and about being a scientist. And I think we have to convey this to students and the general public as well. And the images that I have of science are varied. I mean, Einstein is certainly one, but the other evocative image is being a detective and working with clues to figure things out, or Lewis and Clark being an explorer. And these are images of science that I think resonate even more with me and what I've experienced. And I'll really take the image of the explorer, first of all, because I think really that is what science is. And what explorers do is they start off with a map of where they can, launch their expedition. But they're always moving to something at the leading edge, something that is unknown. And of course, that is the history of exploration. And that is very much what scientists do. You work starting from the known, but you venture into the unknown. And it's that edge that is really exciting and where scientists operate. And I would argue when you're working at the edge of the known, amazing things happen. And often, and you can imagine this from an explorer point of view, that you encounter the unexpected. So it's not this image that think of a brilliant thought and then prove it. It's like you're entering into unknown territory where you don't know exactly where you're going to land. And that is what science is all about. And what I want to share with you is my view of science, that actually the most glorious moments are not those when you prove yourself correct, but when you enter into territory where your pre-existing ideas or the pre-existing ideas of others are overturned, are shown to be wrong by the evidence you collect. And these are like exciting moments in science, and these are the moments that you wish for or hope for, because you're onto something new and exciting and different and interesting. And I'll try to illustrate that, and I will try to share some real experiments with you. I know this is a general public talk, but I'm going to take you through some experiments in this talk. So, okay, my journey as a scientist, just share a few things, and I will go back to some of the early days in Woods Hole, but I grew up in Hollywood. My parents were not scientists. They were from, the movie industry. So science was not part of the background of the family, but I did discover science by doing a high school fair project. And what I learned at that time is how different doing science is from the science I was learning in the classroom, because this is the first experience that I had with actually asking my own question, a question that I wanted to answer and that was motivating to me. And that's the point. It doesn't really matter. I may tell you, and you heard about all these, like, whatever, like awards or whatever. I would just say that I had as much fun doing science when I was in high school as I am doing now. And that even answering a small question and tackling it and attempting to coming up with the answer was thrilling. It was a thrilling experience for me in high school. And, you know, I would say I caught the bug ever since then. Okay, then time goes on, a little bit older. I'm now going to graduate school. Part of my graduate school was working with these individuals, Mike Sheets, Tom Rees, and Bruce Schnapp. And I'd like to tell you a little bit about the problems that we're interested in studying. As you heard, I was a neuroscience student at the time, and I won't go through the whole history, but I became interested in this question, which was how is material transported in nerve cells? Now, this is an interesting question because nerve cells are the biggest cells in your body, at least by dimension. So they're microscopic in size. And in fact, the cell body, which is shown over here, which is located in your spinal cord, you would need a microscope to see. It's like 20 to 30 microns in diameter. But this very small neuron in your spinal cord will extend this enormously long process that can go all the way down your leg, where it then has a nerve terminal that may innervate your muscle, for example, and cause your muscle to contract. So this distance here of this tube could be 1 meter in diameter. Now, all the material that is produced, all manufactured by the nerve, the proteins, lipids, and other core elements, building blocks of the cell are made in the cell body in your spinal cord. There's none of that machinery at the tip of your toe. So the material has to be made by the factory in your spinal cord and shipped all the way down this 1 meter neuron. And at the time, really, no one knew how this transport process worked. But I also want to give you some idea about the dimension of this transport that goes from the spinal cord down to your foot, and put it on a scale that maybe is more relatable on a macroscopic scale to all of us in this room. So imagine an 8 meter cell body. That's the part in your spinal cord that's the manufacturer. It has the DNA. It's making all the building blocks. So 8 meters, like 25, 24, 24 feet, so I don't know, maybe it's from here to the wall, something like that. Anyway, it fills part of the front part of the auditorium and not even the entire part of the front part of the auditorium. Now let's imagine that cell body being that big and imagine where the nerve terminal would be. Okay, well, let's place that cell body where I came from in Janelia. The end of that neuron, if we proportionally create things on this scale, would be right in this auditorium right here. And so you can imagine then the distance of which this transport has to occur. This was a 4-hour drive for us to come here from Janelia. On this scale, the objects that are being transported are pretty small. They would maybe be the size of this laptop, for example. And they would be transported from Ashburn, Virginia down here in about 2 days. So, and you'll see, I think a lot of this transport moves more smoothly than what I saw today on Route 81 as well. So how does this transport work? Well, the transport inside of cells has analogies to the transport that we're used to. And we're used to seeing things move on roadways involving motors. And what I'll show you is that the transport mechanism in cells uses the same kind of strategies. So here's just a brief movie. Here is the transport mechanism we're used to. There are cars moving on roadways. These cars often are carrying cargoes that are building blocks or materials being shipped in the city in different ways. And now if we look inside of a cell, we're zooming in now on what a cell looks like. that being the nucleus over here, you can see structures, these long lines, which I'll tell you more about later, which function effectively as roadways inside of the cell. And on these roadways, you can see cargo being transported along the roadways by machines that are walking along these roadways. And these are molecular motors. So this is the kind of transport mechanism that I'll tell you a little bit more about. So again, I'll try to make this analogy and make the connection of words here. Transport in our human environment involves cargo containers transporting all kinds of goods. We saw a vehicle exactly like this today on the way down here. So there's cargo being carried in these containers. There are motors here that are using gasoline as an energy source to move that cargo, and we need dedicated roadways so that this transport can occur. Now, it's pretty much the same for biological transport as I described. And this movie here shows a cargo. It is a vesicle. For those of you that know, it's made of membranes and lipids. And inside of that container is a bunch of molecules as well. There is a motor that you can see walking along the track over here, and I'll tell you more about that. And it's walking on this extended roadway. So in biological parlance, I will use the word vesicle or organelle as the biological name for what you can think of as a cargo. And these are little containers made of biological materials, as I said, membranes and lipids. There are roadways, and this roadway you see here, in this case, is called a microtubule. It is made of a protein that forms a polymer, and it acts as a stepping stone for a molecular motor, analogy to motor in the macroscopic world. And instead of gasoline, these motors use another high energy fuel inside the cell. It's not a hydrocarbon, but it's the same kind of principle. It's a molecule, a chemical energy source that can be broken down so that one can extract energy, and it's called adenosine triphosphate, or ATP. So what are the types of molecular motors? So there are three major classes of these motors that can walk on cytoskeletal, or what these long polymeric elements inside of cells. One is called myosin. Actually, it was discovered a long time ago in the last century. Dynein, which I'll tell you a little bit about, was discovered in the 80s, and I had the great pleasure to be involved in this journey of discovering kinesin that I'll tell you about. So what I'd like to tell you about is how I ended up discovering kinesin, and also what it is used for. So the real story of this is working right at the edge of the known and the unknown is kind of a messy place to be. You often start with the known because that is what you actually know, and you kind of carry that knowledge into your exploration at the beginning. So when we thought of looking for a motor that was working in the axon that I just described to you about, we thought that a known motor called myosin was going to be the engine that was powering things in the nerve cell. That is how the journey began. And of course, you don't know the unknown, but that journey took us to the unknown, which is a new motor protein, which was called kinesin. So I'll share with you a little bit of how you move from the known to the unknown through experiments and evidence. So our first hypothesis, as I said, came from the known, which was a motor very well characterized, and that is a motor called myosin. It is the motor that powers muscle. It allows the Virginia Tech football team to perform. All of those skeletal muscles that are in motion are powered by myosin. That's what powers your skeletal muscle. It is also what causes your heart to contract. And, you know, this is actually what it looks like, whoops, at a molecular scale. It is a small machine, and it undergoes these kind of energy-driven strokes that work on a filament and cause muscles to contract. So our first hypothesis and our experiment was that myosin was a good candidate, and there was reason to believe this, was the molecular motor for axonal transport. So to test if this idea was right, what we wanted to do was take myosin, coat it on an artificial bead, and then put it in the squid axon. And if it is the motor, we thought, well, it should move along tracks, which are made of a filament called actin, and it should be moving inside the squid giant axon. So this was the first experiment we wanted to do. And, you know, when you do an experiment, you also have to figure out what is the best way to do the experiment, what's the right organism to do the experiment. Most human beings don't want to donate their nerve cells for obvious reasons. So we had to use an experimental organism to test this idea. We chose the squid. And this may seem like a strange choice to you, but there's a good reason why the squid was chosen. And that is, it has an axon that goes from its brain here to its body muscle. And it has one giant axon, which I'm showing you here, that causes the escape response for the squid. So if you've seen a squid like trying to squirt water and get away from prey, that rapid response is caused by this giant axon. This axon is about 50 times bigger in diameter than a human axon, for example. A human axon would look like the diameter of many of these small axons, and you can see how big that giant axon is. That made it a very attractive axon, because we could take now our myosin-coated beads and in this giant axon, inject it into the giant axon and see what these beads would do. So this was the idea for experiment. Seemed pretty good. And these are the results. So what we expected, as I just told you, is we make these myosin-coated beads, we put in the axon, and we expect to see them move, just like the other cargoes. Now, there's another thing in biology, members of the general public, I need to explain to you, which is a control experiment. It's a very important idea in doing experimental biology. And that is to do an experiment almost identical, but remove a key variable to test whether that variable is indeed, you know, the active agent that you expect is causing some result. So in this case, the control was to take the same beads, but now just remove the motor. And of course, our hypothesis is the motor is going to drive the motion. So the control would be, in this case, remove the motor, just put the beads. We've now separated the variables. One situation with the motor, one situation with not. And we would expect no motion there. Okay, that seems reasonable. This is what we got. The myosin-coated beads didn't move at all, and the control beads moved. So when you get that kind of result, what do? Well, you scratch your head. I mean, honestly, this result completely baffled us. Had no idea what to make out of it. And I'm going to come back to this result later. But this is what I am talking about, where the excitement begins in science, not by getting what you expect, but getting things that challenge your view of how living systems work. And this is such an example of the unexpected result. Okay, so what to do next? And well, also, this is a really important point, that science, I should even change can be, is full of surprises. And that is the nature of science. That is actually what you want, because this pushes you to learn new things, to make discoveries. This is very different, I should say, than engineering and medicine, for obvious reasons. You do not want surprises in engineering and medicine. If you build a bridge as an engineer, you want that bridge, to stand. If you go in for an operation, you kind of want to, like, surprise is not like the thing you want to hear from your surgeon or anesthesiologist. So, really the foundation of what allows a bridge to be built for medicine to be done at this level of success is built on earlier science, where people had to learn about engineering, material sciences, or learn how body physiology and so forth. Eventually, the science builds up this foundation to allow this to occur. But I just want to distinguish that as a scientist, you're kind of working at this leading edge of the known, the unknown, and that edge is full of surprises. So anyway, we got an unexpected result. We were scratching our head. So clearly our initial idea was wrong. We needed a new idea. And plus we needed a new approach. And instead of thinking we knew what the answer was, and just going straight there, we now had to let biology speak for itself and give us the answer. And we had to come up with a strategy to uncover what biology wanted us to know. So we took this approach, which was kind of an agnostic approach, to kind of ask what components were necessary for transport. If it wasn't myosin, it wasn't actin, what was necessary? And a little bit of the way strategy in which we did this is kind of illustrated by this diagram over here. Imagine trying to come up to a car, not knowing anything about a car, and it moves, but it has all these different parts. Only some of these parts are really needed for transport. Some of their windshield wipers or whatever, seats, there are lots of add-ons, but you want to get to the core machinery that's really involved in powering the engine here. That's what we wanted to do. So what we wanted to do is take a very complex system apart and attempt to put it back together again and find out, well, this part isn't needed, this part isn't needed, this one's important. That was the general strategy. Now, most cells, including a squid giant axon, have lots of parts. These are also called proteins that make things work in a cell. Proteins are made by genes, and most organisms have 20,000 parts. So it is a fairly daunting effort. But the general goal is to take components of a very complex cell, isolate them, and put them back together and see if one could reconstitute the transport machinery. So again, let me tell you a key assumption we made when we started off on this journey of which parts we were going to try to isolate and put back together and see if they would work. We for sure did not know what the motor proteins were. We knew that there were vesicles. We knew that there were roadways, which after a little while we discovered were microtubules. We didn't know that at the beginning. But we thought also the motors are going to be attached to the organelles. I mean, it makes sense. In a car, the engine's in the car, right? So pretty natural assumption. So we assume that if we isolated the organelles, they're going to have the motors attached to them. So our goal with this experiment was to first isolate these cargos, the membrane organelles. We can make microtubules by isolating the protein, and we can buy ATP from Sigma, and we were going to recombine them together, and voila, the transport system would be reconstituted. So that is this first experiment shown here. This is what an experimental scientist does. They have little tubes with stuff in them, and they take this little device, which is called a pipette man, and you pipette little liquid together, you mix them together, and you put them under a microscope. So here you had a tube of microtubules, ATP, that's the energy, the organelles, and we recombine them, put them together under the microscope, and it's going to work, right? No luck. Nothing. Nothing. Okay. Rather disappointing. Took a long time. This sounds really easy. Even this took months to actually get this far. All right. So then back to the drawing board. Well, maybe we weren't so smart. So we now had to think of, okay, what else might be needed? If that wasn't enough, something else might be needed. So we took basically a soluble fraction of likely 10,000 different proteins in a mixture, which are called soluble proteins, and now added that along with the organelles, ATP, and the microtubules. Whoa, this worked now. Very exciting. And this is kind of what we saw. So there are these roadways there that you can see. These little objects are the microtubules, and you can see that they're moving along really well. So this was a really big moment, very exciting. But now remember this thing I called a control experiment where you leave out a variable? Well, that evening, same evening, I left out a variable of taking away the membrane organelles. I wanted to make sure that those little objects that we saw moving were actually the membrane organelles that we isolated, and were not some particle that came around from the microtubules or whatever. It was a kind of experiment, just, dotting the I's, crossing the T's, making sure, everything was right, being very diligent, and with no intention of seeing anything here at all. But then you can say you tried that control experiment. Well, what happened here was like unexpected, and I will use the word mind-blowing because it was mind-blowing. But I'll just go in with the expectation, expecting to see nothing, quick 10-minute experiment, go home. What happened was that the roadways themselves under the microscope were moving along the glass cover slip. So if you just follow any one of these points over here, you will see that these roadways, these microtubules, are moving along the glass. This was totally anticipated. We actually, watching this, it took a little bit to even think what in the world was happening here. Until it came to me that there was another way to interpret this, and I'll explain that in a second. And I'll come back to that assumption that I made, that all the motors were on the membranes. I told you that was our assumption going in. That was wrong. And I'll explain why that assumption was wrong and why how these microtubules are moving. But first, let me show you a phenomena on the macroscopic world that you may be more familiar with, and that is crowd surfing. So this is Bruce Springsteen here, but it could be a number of other people. And you can see that the boss here is being transported across the crowd, and he's just hanging out there. And there are all these little arms here that are passing Bruce from one arm to the other, across the audience here. Well, what is happening here was effectively molecular crowd surfing. Think of these microtubules here as Bruce Springsteen here, just hanging out. And underneath these microtubules are a whole bunch of little hands, the motors that are attached to the glass. And this is the model of what's happening here, that these motors, for whatever reason, first of all, were not all attached to the organelle. That was incorrect. A lot of them were floating around free, and they were in that soluble mixture, that unknown mixture that we got organelles to move. There were a lot of free motors, they would not only attach to organelles, but they would stick onto surfaces like this glass slide. And that is what caused that phenomenon. Now, let's go back to our earlier experiment. Suddenly, like a year later, there was an aha moment of how to interpret that experiment. Because I mentioned here, myosin-coated beads did not move. Sorry, the control beads here, the surprising result was that they moved. The myosin-coated beads didn't. And now the interpretation was these free motors that were around could stick onto these control beads and get them to move. And that's what these beads moving look like. The reason why the myosin-coated beads didn't work is that the beads were already coated with something, and these magical motors from the squid giant axon couldn't attach to them and couldn't move it. So now, all of this really came together into this kind of model, that there was some kind of motor. Yes, its biological function was to transport vesicles, but there was kind of a reserve pool of these motors. Not all of them were attached to the cargo. And these reserve pools, of course, they never have seen a plastic bead before, or they've never seen a glass slide before. But fortuitously for us, these reserve motors would also stick to these surfaces and get them to move. So effectively, what we were able to do is to trick the biological transport system. Normally, there are these natural cargoes that are moving inside the axon, but we could create a situation where these motors would now transport artificial things that we put in the system. And honestly, this was a giant breakthrough for understanding how these molecular motors work, because now we can get these motors to behave, perform for us, do whatever we want under kind of our command and our control with these artificial surfaces and plastic beads. And this system led us to discover what that motor protein is that is sticking to that bead. And that was this motor protein that we ended up calling kinesin, kine for Greek for movement. And using systems like I just showed you, the microtubule crawling on glass, the bead moving along the microtubule, in the next 15 years, could really study how that transport worked outside of a cell in a test tube. And I can't go through this entire story, but we learned actually what this motor protein looks like. This is actually a very accurate depiction, at least of the shape of the motor. We learned all the dynamics of how it uses ATP, we and other people in the field. And we learned actually how it walks. And it walks, it's a two-headed molecule, and it walks in this hand-over-hand manner where the front head, and if you watch this red thing over here, it pulls the rear head from this backward site to this forward site on the microtubule, kind of like a judo expert that can flip an opponent from rear to front, and it leapfrogs steps along the microtubule that way. So this motor is pretty amazing. It's, you can see it's 100 millionth of a meter in size, very, very small. I told you it uses ATP instead of hydrocarbons. It moves at a few millimeters per hour, which sounds really slow, unless you really think about how small the motor is and how many lengths per unit time this motor is moving. And it moves actually faster than, a Porsche on the freeway. So, and the other amazing thing about these motors is they are more efficient even than your Prius or Tesla. They work at about 60% efficiency in converting chemical energy into motion along the track. So they're really amazing examples of biological machines. So, we can also ask, well, that's very nice. Are these motors important? All I can say is I started off with this slide, motion is essential for life, and I really mean that. even when life starts, the act of fertilization involves sperm swimming to the egg. All of that swimming involves molecular motors. The act of cell division involves molecular motors. That's a physical process. A lot of the processes of the entire embryogenesis and development, again, involve motors putting materials in the right place in the developing embryo. And I showed you that example of DNA division. In fact, they are so fundamental for life that most defects in molecular motors, if you knock many molecular motors out, you will not have a viable embryo at all. That said, there are a number of genetic diseases that involve smaller perturbations, point mutations in molecular motors that do cause many diseases in humans. This is just an example of a foundation that is trying to develop a cure for a Kinesin motor that causes neuropathies, defects in the nervous system in children. There are about 45. What I told you about is one Kinesin motor. Since the original discovery of that one Kinesin, we now know there are 45 different Kinesin motors. The reason why there's so many is that they perform many different functions inside of the cell. So this genetic disease involves just one of these 45 different motors. We now that we understand how molecular motor work, we have the opportunity to think about treatments for some of these diseases. This is just an example of a disease of the heart, basically called heart failure, where the cardiac muscle does not contract as strongly as it should. This is a very prevalent disease, and a company which I will disclose I was a co-founder of has been working to develop drugs that work directly on the motor cardiac myosin that is responsible for pumping the heart. It actually makes that myosin perform better than normal. It activates the motor, and this just shows an echocardiogram of a patient here with heart failure. This is left atrium and left ventricle, and this is the mitral valve here, and you can see it's kind of fluttering. That's abnormal, but after activating the myosin, you can see this the contraction of the heart much stronger, and you can see this valve popping. And now this drug is right now in front of the FDA, actually this December, for approval for this disease. So what I would stress is all of that work at the leading edge of the known and the unknown, all of that had to happen before one can even think of making any new drug. And that's just an example of one that I just showed you. But this, when you think of any drug you take, all that pill is made possible by a background of basic research and curiosity-driven discovery to figure out what that drug is, how to make a drug, what protein to target, how that drug should work. So now I'll just briefly advertise my talk for tomorrow. What I told you about was work on vesicular transport. Maybe that goes back to the roots of Bonaventura Corti. Tomorrow I'm going to talk about the motion of cilia and flagella. So going back to the roots of Van Leeuwenhoek. And again, cilia and flagella are very familiar things. They are present in your respiratory tract. There are these little arms here that clear particles out of your lungs when you get sick. You're also probably familiar with sperm. They have long flagella, and that's how they move to find the egg. They actually swim. And what I'll tell you about tomorrow is this, again, amazing motility engine that's even much more complicated than the one I just described. I'm just going to show you this movie of the structure of what this thing looks like, just as a teaser. But you can see it's this incredibly complicated machine inside of these Cilia and Flagella that are making these structures beat. And we're beginning to understand more about how this machine, which is made up of about 1,000 proteins, work. So in the last just very couple minutes, I'll tell you about my other passion besides science, besides doing science, which of course I love, but that is once you make a discovery, that discovery is only useful unless you communicate it. And the communication of science to other scientists is very important. Communication of science to students also builds the next generation of scientists and a general understanding of what science is, even for students that do not decide to go into science, become, part of the public that needs to understand the scientific process as well. So I do think that knowledge of science and science education should be a global public good, and that's our responsibility as scientists. So you heard about this one project that Karen and I started a number of years ago called iBiology. This was to make videos, even in the early days of the internet, publicly available, so that you don't have to get a speaker here. You can actually go, anyone around the world or any institution around the world can view seminars from leading scientists. This is a new project that I'm working on that I'm very passionate about. I love if there would be some interest or dialogue with Virginia Tech, but it's called the Explorer's Guide to Biology, and it's a free resource for undergraduate education. And it's effectively trying to a little bit rethink how we are teaching science. And a big premise of this project is to teach science a little bit the way I've been engaging with you today, which is to understand biology through stories of discovery. So, there are traditional textbooks that they operate through a book format. This Explorer's Guide to Biology, XBio for short, is on the internet or download. Textbooks can be $300 a piece. XBio is free. This is my opinion. And I think, the approach we've been thinking about biology education as a sea of facts just is not working anymore. Also, there are just too many facts in biology anyway. It's more important, really, I think, that people understand science as a process of uncovering knowledge, not just the knowledge itself. And I think that's how we have to steer science education overall. So in XBio, you can hear about the story of motility. Yes, you can learn about Kinesin, but students can hear about this story through this narrative told both in written form, in beautiful illustrations, and also with videos in XBio. Or you can hear about the CRISPR, which changed the world, through the words of Jennifer Doudna, as she uncovered CRISPR and its use in genetic engineering. And there are a bunch of other great authors. Many of you may not know them, but I can assure you this list here is full of Nobel Prize winners, NES members, or people that have made major contributions in science. And this is just the start of the project. This is really going to be a 10-year effort to build up this whole complete narrative of the life sciences. But we're on this journey, and I have some cards here if anyone wants to see some of the illustrations and wants a card afterwards. But they're really cool and beautiful. Lastly, Janelia Research Campus. We're your neighbors. Come visit us. Who we are a research campus of the Howard Hughes Medical Institute. Howard Hughes Medical Institute is the largest life science philanthropy in the USA. We're your neighbors in Ashburn. And what we do is, I would say, invent and discover. It's an interesting place where probably half the scientists are working on technology development and half of them are working on curiosity-driven, question-driven discovery. And it's also an unusual place that really aims to foster a really high degree of collaboration between science as part of its framework. What we do right now is we study the brain and the body. A group of scientists really trying to understand how neurons compute. And there's a new group of scientists coming who are trying to understand basically how cells and organs also do their computation. And there are a number of tool builders, technology developers. Particular strengths are in imaging and developing probes for imaging and in computation and theory. And, Virginia Tech is producing all kinds of fantastic people here. So I would just encourage an awareness of everyone that we are in the neighborhood. We are constantly offering jobs in engineering even, and computation as well as biology. HHMI is deeply committed to diversity with major investments in this, even in recent years. I encourage you to look at HHMI for all of its efforts, and come look us up. That's the main thing. And thank you for the invitation here, Mark. Thank you so much for sponsoring this lectureship, and Daniela for the invitation. And I'm happy to answer. It's getting late, but I'm happy to answer a few questions. Remember, questions in biology are important.
I think the other one's working. I'll go over there. So then.
Or you can come up here. You can.
All right. Should I just ask them? So I was curious. Like in the beginning, you showed a video of like a huge globule, right, and like a teeny tiny motor pulling it along.
Yeah.
So I was wondering about the strength to size ratio there. And I know like things are like really small, right? It's all different. Like insects, right? They're very tiny. They can go really far. But I was wondering about it from like a biological point of view. Like, is there a reason for that strength to size ratio, like coming from biology, or is it just because these molecules are so strong that it doesn't matter, they can be as strong as they want to be? I don't know. Does that make sense?
Yeah. Well, basically, first of all, maybe there are two things in your question. One is like, wow, that the motor looks so small, and that, that vesicle looks so big. Does it even have enough, power to move that entire big object? If you do the calculation, actually, it's a little counterintuitive. If you do the calculation of the amount of force it requires to overcome like the viscous drag, so like pulling this object through an aqueous environment, the viscous drag from even this much larger vesicle is fairly small, at least in a test tube in an aqueous environment. So so it is a little bit counterintuitive, like in the microscopic world, how much force is needed to move these organelles. Now, that said, that calculation, as it's usually done, is imagining something moving in water. And the inside of a cell is a very different environment, and it would almost be called non-Newtonian, because the viscosity is not just made up of fluid like water, but if you look inside the cell, it's actually a jungle of filaments and other objects. So it's more like dragging something, not through the street, but through a really dense tropical rainforest. And so probably, first of all, these motors have to be stronger to, overcome like a lot of the other structures that are found in the cellular environment. But, the other, answer is an interesting one, that there are, as I mentioned, there are 45 different motors. And they carry different things. So you can ask, why are they different? Well, they carry different things, but also interestingly, a lot of their biophysical properties, such as the amount of force they produce, the speed at which they move, um, those are the main two parameters, can differ considerably from one to the other. And we really don't have a good idea of why one motor is moving at that speed and has that force versus another. So there must be evolutionary reasons that work on the biophysics of these motors and, exquisitely tune these parameters. But, that's an area that's still like open for understanding, I would say, of, how evolution is actually tuning the physics of these motors. We still don't completely understand that.
Looks like we have a couple questions on what people have been.
Oh, can I see him? No.
I can ask the question while she finds it.
Oh, wait, I can see him now. I found him too. Okay, go ahead.
I can ask the question while she was waiting.
Yeah.
It's not really a specific science question, but I'm in my second semester of the PhD, so just beginning my career. And you have done so much advancements in science, but you've also done so much in outreach, giving back to the community, which I just think is so fascinating. And I was wondering if you had any advice for a first-year PhD student on how to do both of those things.
Yeah. So the question, I don't know if I need to repeat it for the Zoom audience, but yeah, the question was, I'm sorry, what is your name?
Rebecca.
Rebecca is a second-year PhD student, just kind of getting started on the research angle of things, just kind of getting into experimentation. And how do you balance the research with all these outreach activities? First of all, I would say a lot of the things you heard about that I did came much later in my career. That's point number one, where I felt like I had a little more room to do that. But what I would say is I actually encourage, personally, PhD students and postdocs to get involved in outreach for a couple of reasons. It doesn't have to be at this like gigantic, massive level of starting a nonprofit organization, but whether it is working in schools or, giving talks to the public or whatever opportunities there may exist. If there is something nourishing and rewarding for you that actually gives you pleasure and purpose, that is very important. And I think you need that grounding in really things that are meaningful to you to also do well in research. And honestly, in many cases, the amount of time required to go into a school and teach kids is such a tiny fraction, even of the amount of time you spend doing research. But it can be so gratifying that I mean, that's my view. I've always encouraged my PhD students and postdocs. I do hope that a lot of senior faculty will also, adopt that view too. And the other reason why this is also important is, we are seeing in our society also, I think, a need to get out of the ivory tower. I think science has operated in a very, closed environments of universities or institutes for a very long time. And, we're seeing this come to roost right now, where a lot of people, in society overall really have never met a scientist, don't have much understanding of science or even fear of science right now. And I think unless we get out and engage with people at all levels, I mean, PhD level too. In fact, I think oftentimes PhD students and postdocs are better ambassadors of science to the public because, they can see this is a passion of yours. This is what you want to do for your career. it's a very powerful message, and in some ways different even than when I go out, and speak. So I'm not sure I totally answered your question, but also, yeah, the other thing is start small. And the other thing is no effort is too small. That's the other thing. Like, it all adds up. And what I've discovered, later in my life is I really enjoyed it. It actually gives me a great deal of joy and pleasure to have this outreach element of my life, as well as the science. And I think they're both creative too. That's the other thing that I've learned. So. Should I read some of these quickly? Well, Priyaka asked something I think that was very similar to what the other gentleman asked. Thank you for your talk. I really enjoyed it. I have a question. How heavy is the vesicle? Can the motors carry heavy load? So that's a little bit like the question I answered is A, they're not very heavy. And it's not gravity that's really the factor or the force here. It's really the viscous drag and overcoming viscous drag. But even that in an aqueous environment is very small. In cell is probably much larger. All cars, this is Stephen Melville, all cars need a driver for directionality. How do vesicles know where to go? Oh, that is an excellent question. And we are working on that because you're correct. Now, in the case of the microtubule, there are two kinds of motors, and I didn't go into this. Kinesin moves in one direction, dynein moves in the other. So it's a critical choice. If you pick up a kinesin, effectively you go to the tip of your toe, the nerve terminal, pick up a dynein, you go in the other direction to the cell body, and that's a 2-day journey. So if you make a mistake, that's not so good. So there are like regulatory mechanisms and cues and signals that put on or activate the right motor on the right cargo. This is still like a really open question right now. We have some, I can't go into all the things. We're making, as a field, progress with it. But actually the one project I've taken with me to Janelia is working on this very question. And to answer this, we've actually created, again, not a bead, but kind of an artificial cargo that we can put in cells, and are kind of learning how to switch the directionality of transport of these artificial cargoes. And we hope that will give us some insight. Kulyash, as a leader of biological research, what are the next big things we should expect from cell biology? What fields of research look promising to you? Well, I often get that question. There are two things to say. Every field is amazingly exciting in the life sciences. I mean, what I would say, when you read a textbook, it seems like everything's known. My view is we are just scratching the surface of understanding how living organisms work. I mean, honestly, we can't explain how a cell works, let alone even the simplest organism like a worm. The brain is a mystery. I mean, we are very far away from understanding many of the fundamental rules of how living organisms work. As a field also, I think the biggest challenge is also the amount of information that we're getting. I've showed you some very simple experiments, yes, no, kind of framework. We're now getting data from experiment may have, data from gene expression of 20,000 genes, over 20 time points, or 3D data sets of images that are massive. And, one of the real challenges in biology now is how to process all that information so that human beings can understand that and derive knowledge. I think that's one of the exciting frontiers. All right, Zachary asks, hello and thank you. You talked about transport with microtubules for future studies. Could this relate to targeting for search and medical uses in relationship to transport? How might that look if possible? Yeah, there have been some thoughts that maybe you can put drugs on, for example, at the periphery of the nerve system and have that drug be transported or CRISPR agent or whatever back to the cell body. So there are some possibilities in that, have not been realized yet. I'll ask these two, they're really quick, and then I'll ask, the role of science and scientists has changed, stay there, from the time of Leeuwenhoek. How have the roles and responsibilities of scientists changed more recently, say in the last 20 years? Well, I think I partially answered that question. You know, I think A, we have to communicate better, and B, we are dealing with some very powerful technologies globally, you know, whether it's artificial intelligence, whether it's CRISPR editing, our tools are a lot more advanced than the times of Leeuwenhoek. Is there a role for AI at the edge of discovery for a different aspect or insight? I just talked about that. That's Paul Thorne. You know, yes, for sure. You know, how we deal with these very, very large data sets, I think is going to require, you know, new tools for artificial intelligence. I'm going to talk about this tomorrow, but DeepMind produced this program called AlphaFold, which is an artificial intelligence program that I'll share with you some tomorrow, has revolutionized structural biology. It is just came out like last year. It's probably going to win the Nobel Prize, maybe not this year, but if it just won the Breakthrough Prize this year, which is a massive prize. It's clearly going to win the Nobel Prize. Probably should be careful what I say, but it is a massive revolution, and it came from AI, and that was the big breakthrough that they made. All right, please, and maybe the last question, or two more questions.
So myosin has been in with the other transport that we knew about, right? Before Kinesin.
Yeah.
So how similar are they? Are they only like superficially similar in that they look like two bodies that are sort of moving on top of each other, or would they perhaps could one have been adapted from the other early evolution?
Oh yeah, wow, that's a great question. That was a major part of my work, I don't know, maybe 20 years ago. And you're dead on right that, you know, originally people thought that myosin, I mean, I'm talking about all scientists, including myself, there was no most scientists, based upon more evidence than I have time to describe here, thought myosin and kinesin were totally different. They had no relationship. They work on different tracks. The roadways are different. Computers looked at them and found no amino acid homology. So that's like the sequence of letters that make up the, the string of amino acids that make up the proteins. Computers found nothing. Then we got our lab with Robert Flederick's lab got the crystal structure of kinesin. And then that was also one of those mind-blowing things, because we found that the kinesin structure in 3D, not in linear amino acid sequence, but its 3D shape and look and exactly real details of the way it looked overlapped between kinesin and myosin. And it was very apparent that kinesin and myosin were more closely related to one another than either one was to any other protein ever made. So probably what happened is that there was a very ancient motor, you know, we don't know what it was, probably disappeared, that was a precursor, and it kind of split off through evolution and created the myosin lineage and the kinesin lineage. So yeah, that's a great insight that you had there. And frankly, it sped up our research in kinesin by years. And the reason is people have been studying myosin for decades. And the finding that this was like a cousin of myosin allowed us to kind of see parallels between the two motors and things that were similar and maybe some things that were different. But it really bootstrapped our ability to understand kinesin. But yeah, that was one of, again, those amazing, like, wow moments when we discovered exactly what you said. Might be true. Yeah.
So I know I'm extrapolating a little bit from like my understanding of how experiments work, but I imagine now a great deal of funding, effort, went into your first experiments with the myosin-coated beads and the squid models, how did you redirect energy after that experiment failed or you received unexpected findings?
Yeah, so it's a great question. So, like you invest a lot of time and, almost like with all that sunk effort and it didn't go anywhere, like whatever, what do you do next? Well, I think there are a couple important points. I'll address it from several points of view. One is your mental energy, like, oh my God, nothing's worked, right? And just to keep going. So that's not funding, but that's an even more precious commodity, which is your will and motivation and drive, right? And this is the real difference. And when I said you're at this boundary between the known and the unknown, and biology nature is not like a biology exam. You don't know how long it's going to take, or you don't know when you're going to make progress sometimes. So, there are times, and this is good for PhD students to know, this is the main thing you learn as a PhD. There are times when it seems like things are not advancing, like you're not getting the result, you're not learning, you haven't got the answer, whatever. You're still making progress, because every time you learn about something that doesn't work, your thinking is going in a new direction. So that's one thing. Resilience and patience is an incredibly valuable commodity of being a good scientist. It's much more important than being brilliant, than like Einstein, in my opinion. But the second thing to know is that time is very nonlinear. If you're on this really slow slope, seems like it's not going anywhere, you kind of extrapolate, oh my God, this is going to be the longest PhD thesis on Earth, the way this is going. But science isn't like that. Those experiments, I went from like nothing happening for 6 months. Some of it was like I screwed up experiments. I did, I messed up, there's not just the ideas, but you also sometimes just fumble stuff in the lab even. I went from 6 months like nothing. And then I showed that movie of that, the organelles moving and that success slide. That happened after 6 months of trying. But the next experiments, the microtubules, the beads, a whole bunch of other experiments all happened within 3 days. So, sometimes science is very nonlinear, meaning like it feels like you're in a rut, something clicks, and then you're on this totally different slope. Regarding funding, that like at Janelia particular, we take, for example, here we have a very long-term view that if you're going to try to do something hard, there may be long spells when nothing's happening, and you still got to stick with it and, have that patience and determination, to get to the very end. But as a student, I think the most important thing to learn is this element of resilience. And it's also something I would say, it's very personal. You can listen to me and like, okay, Ron's telling me that, but once you experience it once in your life, then you understand it, and it like changes your perspective of doing science and being a scientist ever after that. So particularly for when you start in science, like stick with it, don't get discouraged. Go work at schools, go talk to friends, whatever. And just understand that sometimes it's not you that's not getting the answer or not making the experiment work. It is the nature of doing science. You can't personalize it either. It's just, part of the nature of tackling the unknown. And I think that's an important perspective too.
Thank you.
These are great questions.
Good evening, everyone. I'm Bob Cohen, Department Head of Biological Sciences. But just a few brief comments to wrap things up. First, I'd like to thank Ron for an absolutely fantastic talk and generosity with your time. As you heard, we'll also be giving a more specialized seminar tomorrow at noon in the Braylon Auditorium. I don't know if you know this, but I've been following your work and your work with your former students, postdocs for nearly 3 decades. You must have been a teenager when I started. But it was great to have a front row seat to hear how all these great discoveries unfolded and to hear your perspective on scientific discovery. Huge thanks also to Mark and Debbie Sowers for your continued support with the Sowers lecture series, to Janelia and Janelia for nominating you and being your host. Lastly, I'd like to thank Dean Pitt, Jenny Orzalek, and the entire team staff for their support. Finally, I'd like to thank the audience for coming out and tuning in to tonight's talk. And just a quick announcement that the next J. Mark Sowers Distinguished Lecture is scheduled for March 1, 2023. The speaker will be Dr. Matthew Jackson, who's William D. Everly Professor of Economics at Stanford University and game theory expert. Please keep an eye out for additional information as that get here. I look forward to seeing you then. In the meantime, have a safe trip home and great evening. Thank you.